Hello, and now we're coming today from Australia, and I'm very, very fortunate to have Professor Eva Dimitriadis, who's an absolute expert on decidualization and implantation and has done a lot of research for quite a long time, who works at the Royal Women's Hospital in the research department. So Eva, maybe before we start, if you can tell me a little bit about your career and what you did in terms of implantation, but also why you did it. I think that would be really inspiring for a lot of people.
Yes, yes. Well, I did my undergrad I studied biochemistry, and then I really started my career in reproductive biology as a postdoctoral fellow. I did my PhD overseas at Trinity College in Dublin, and then I returned back and I got a postdoctoral fellow position with Professor Lois Salamonson at Prince Henry's Institute here in Melbourne. And Prince Henry's Institute was affiliated with Monash University, and I started working on the role of cytokines in implantation, and initially for contraceptive purposes to see how we can block implantation for contraceptive purposes. And then I just became fascinated with the whole process and implantation and how there was very little known about it, particularly in humans. So I developed my own team, and I progressed basically really to understand maternal fetal interactions and how pregnancy is set up. So that's it's a black box area because obviously it's very difficult to study in humans. So my work really has focused on setting up appropriate in vivo models and ex vivo human models where we can really model the early stages of implantation to try and find out what is going on and then also what is going wrong, basically, in infertility, in pregnancy loss, recurrent pregnancy loss, and also other conditions of pregnancy such as preeclampsia and fetal growth restriction. So what happens early on really determines the health of the pregnancy as well.
So do you want to explain a little bit about what decidualization is and why that's important in the process and some of the biology behind it, really, so that we can help explain that to our patients? Is that okay, Eva? You're
Yes.
probably the world expert on this, so.
Yes, absolutely. So it's a process that occurs in the uterus, and it's really the morphological and biochemical differentiation, so the changes that occur in the endometrial stroma. So to prepare the uterus for implantation. So this process occurs under the influence of progesterone that's released following ovulation, because the endometrial stromal cells, like, they express progesterone receptor. So progesterone mainly acts via these cells, and then these cells influence all the other cells around them. So if progesterone is not acting appropriately on these cells, this really the tissue becomes very dysregulated, and it's not a hospitable environment for an embryo to implant. So it's really a critical process in the luteal or secretory phase of the menstrual cycle.
Eva, they talk about the window of implantation and what's different biologically of that window compared to either pre-receptive or post-receptive endometrium. What are the kind of biological differences of that very special time, and how does progesterone have a role in that?
Yes, well, as I've mentioned, progesterone must act on the endometrial stromal cells. And then once they are decidualized, then they interact with the surrounding cells. So basically, what happens in the receptive phase is that you have these functional and morphological changes that occur to the endometrial epithelium that lines the uterus. So that's the point of embryo attachment. So those cells really become quite different. Normally they're refractory, so they do not allow embryos to attach. But in the receptive phase, they have to change considerably, and they put down an glycocalyx that's not there in the other phases of the cycle, so embryos can attach. And then, but around the same time, the endometrial stroma decidualizes. So that's really important. And then the underlying stromal cells, they interact with the epithelial cells to really facilitate embryo attachment and then invasion. And also the endometrial fibroblasts really provide an immunosuppressive kind of environment for the embryo, so it's not rejected by the mother's immune system as well. So that's really important. So the endometrial fibroblasts, or the stromal cells, really are responsible for creating this immunosuppressive environment, so that the fetus is not rejected. So it's really quite a critical process. And until recently, so now we're using technologies such as single-cell RNA sequencing, where we can sequence every cell in the endometrium, and this has been done throughout the menstrual cycle. And information now has come out that really decidualization was thought to occur probably in around the receptive phase or the late receptive phase. But now with the single cell analysis, we can see that it starts very early on in the luteal phase, and then it kind of progresses. So, that's new knowledge that we didn't have available before, and we know what molecules change and what really is required for appropriate decidualization, and then also allowing embryos to implant.
Fantastic. So obviously that's a very complex process. How does it go wrong? Do you have some ideas? And I presume there's not just one thing that can go wrong. There may be different aspects to that. How do you investigate that, and how do you work that out?
Yeah, well, things do go wrong in the decidualized stromal cells and also in the epithelial cells. And I've done lots of investigations with both of the cells. You get epigenetic changes. There are changes in non-coding RNA, in other, things like acetylation. So these are epigenetic changes that occur. We know that, and that could be the result of environmental influences, like many environmental influences, such as environmental toxins, for instance. And, there's many other, psychosocial factors. Like we really don't know what creates it, but we know there are environmental influences. And we can see that if we take a luteal phase biopsy, and then we look at women who are infertile, or even women who develop pregnancy disorders such as preeclampsia, we can see changes in the endometrium already pre-pregnancy that are associated with women who develop these conditions. So, and some of these factors, because the decidualization starts in areas surrounding vascular cells, we can measure some of these factors also in serum. And then some of these factors are also released in uterine fluid as well. So we could potentially sample uterine fluid or serum factors where we, down the track, although this hasn't, there's no tests available like this, yes, but down the track we can potentially look at non-invasive ways to determine whether the endometrium is adequately prepared for implantation and whether decidualization is appropriate as well.
There are some commercial tests, things like the ERA test, and also microbiome tests. There's quite a few different tests. How, from a biological perspective, and someone who does a lot of research in this area, how do you know? There's a lot of controversy as to whether they work or whether they're valuable or whether they're economical. What do you feel? Do you think that's a step forward along a progression to better testing, or do you feel they're worth it? Or, do you have a view on those tests?
Yes, well, I think it's, there's been a lot of conflicting studies, and I think what we have to be very careful to determine which women these tests might be applicable to. And I think we're not there yet, basically. So these tests are quite often done as a last resort when nothing else has worked, So that might not necessarily, work for that woman as well, just doing it in that kind of, as a last resort, basically. So I think, potentially there could be women where it could work for, but I don't think we're at the stage where we can determine which women would benefit. The microbiome studies are really interesting also, but there are problems with sampling. It's very easy to contaminate samples, for instance, and then to determine what's real, what's coming from the uterus, what isn't. I think we still need to do studies. There are other tests that people are doing for other gynecological disorders like endometriosis that I'm sure, which is your expertise, where people are looking at things like, saliva tests or other, testing other bodily fluids. But I think, I think the microbiome is really interesting, and it potentially does, impact receptivity and implantation, but I think we don't have the data yet. With things like the surgical tests, like the ERA test, and there's several other tests available, I suppose with that, you need to take the biopsy at a previous cycle. Sometimes it's a natural cycle, other times it's a progesterone stimulated cycle. And then we know that, well, for my work and many other people's work, is that there are there's dysregulation of progesterone receptor, for instance. So giving a woman, say, progesterone, it might not necessarily work to prepare the endometrium, particularly if the receptor's not present. And we know that in some women that, the progesterone receptor is actually not there. So you can give that woman as much progesterone as you want, and it might not act appropriately, on the endometrium. So I think we have a lot of work to do there. And although it might work in some women, it might not work in all women. So going back to the biopsy, you have to take it, in a previous cycle, and then you have to predict, in the next cycle, or even, we're told 6 months down the track that we could, do an embryo transfer. Now, there aren't many studies that have looked at variability of the cycle, between months. So there's very small studies. It doesn't take things like epigenetic factors into account. So I think, although, it's interesting and it might work in some women, I don't think we can predict which women it might work with, because there's too many variables currently, basically. So yeah. So ideally we would have a test that's not invasive that we could do in the transfer cycle. And there are potentially, like a blood test or sample uterine fluid. That would be ideal to do it in the same treatment cycle.
So at the moment, the research, where is it heading in this area? Because I know that it's really changed a lot. It was almost impossible to get any information in this area until we had good models, and people have really made a lot of progression, but we've still got a long way. I think there's a huge potential to find out a lot more. where do you see things going in terms of research directions? And also, I guess in 10 years' time, what do you think we'll be doing? Do you think we'll be making it much better for people, is I guess what we're hoping?
Yes, well, I think we are. I think with the single cell analysis now, we're really starting to tease out, the molecular mechanisms, at a cell specific level, which is really providing us with a lot more information as to what establishes receptivity. And then we could potentially develop targeted therapies for that. And also, whereas previously all the microarrays and all the information that we have had over the last 20 years has been on whole endometrial tissue, basically. And that's a combination of all the different cells. So say if I was interested in looking at, how an embryo attaches to the endometrial surface, now I can get information of how the endometrial lining is dysregulated specifically, because unless you have the firm attachment, you will not get implantation. So that's really the cause of implantation failure. The embryo does not attach. So now we can really see what happens at that particular cell. And then we know that, because that's directly adjacent to uterine fluid, we could potentially sample that non-invasively. And then we could do things like, do targeted treatments. Say if there's a dysregulated factor, we could inject it into the uterine cavity at the time of embryo transfer. So now that's done with factors, say like, beta HCG, but it's really non-discriminate. It's not personalized. So I think, we are getting to the personalized approach. There's also things like the cell-based therapies. There's a lot of people looking at, cells and stem cells, and potentially we could look at the stem cell based therapies that could, facilitate the endometrium to be receptive. So I think that's an exciting area as well.
What about AI? Do you think that some of this data will personalize things through kind of these large network analyses?
Yeah, like AI, yes, basically. I mean, we could potentially, AI is already being used to determine embryo, health, I suppose, so that's really exciting. It could potentially be used for the endometrium as well, although it's not really, being used yet. Yeah, a lot of work needs to go into kind of, I suppose, just annotating if we're going to look at it in terms of not using, say, the noise criteria that we use now, where a pathologist has to look at the endometrial biopsies to see what stage of the cycles they are. We could potentially do that using AI. But currently there's not many people looking at the use of AI in terms of endometrial receptivity, but certainly if you do develop, say, a biomarker test, certainly AI would be useful in that, for sure. We could use it for biomarker testing, absolutely.
Well, I think our time is up, Eva, but it's been a fantastic conversation. I feel like it's way too short. That's the only thing. And I really, really appreciate all your expertise and just the way you can help us picture what it looks like with the endometrium. I think, and what that embryo is seeing, really.
Yes.
And obviously, it's been a lifelong work. I'm sure there are many more exciting avenues that are ahead of you. And I'm sure we'll hear more in the future as well. But I would just like to thank you so much, because just being able to share your expertise with Aspire across the Asia Pacific. It just makes such a difference to young clinicians and young researchers who may well have a great interest in this area. And I'm sure that you're going to get some questions and potentially people are reaching out to you just to find out a little bit more about this area. So I'd like to thank you very, very much. And I very much hope you can come back and do another podcast on another day, and we can go into more detail in some other aspects of this really fascinating area, which is kind of the last frontier for us. So thank you very much.
Yes, thank you. And I'm very happy for people to contact me. There's obviously a lot that I didn't cover, all the new models and everything that we can, model receptivity and implantation in vitro. And thank you very much. And for all the work that you do, you're an inspiration to me as well. So thank you.
And vice versa. So thank you.